Hello, can somebody please confirm that you can hear me?
I can hear you.
Great, thank you. Okay, are there any questions? If not, before I show my worksheet, I've got the grade book open to tell you about what I've graded. I know I've graded the extra credit quiz. Where's the mouse? Oh, there it is. I just can't see it. And I've graded quiz 1B. And I also graded, not graded, but I gave you the extra credit question on quiz 1B is the one you got the extra credit question for. So everybody got 3 points for the extra credit question. And then quiz 1A I have graded. That's what I meant to say. And I also graded the lecture worksheet for the last lesson, whatever number that was. I've graded the extra credit picture and the extra credit study sheet. Extra credit study week 2, meaning your group studies, is graded. There's 2 more things, just a minute. And for most students, but not all students, I tried to get it finished, but I did not. I got it finished for most students. I've got lab 00 and lab 01 graded the second time. There are 3 students who I have not graded lab 00 and lab 01 the second time. And I'll try and get that done tonight. No guarantees, but I'll try. There's one more thing I need to discuss. The current 2 labs, lab 4 and lab 5, I've got them graded for the first time. Make sure you can resubmit answers to those. Yeah, you can submit answers to lab 4 until June 30th. That's this Saturday. And I make sure that's true for lab 5. Yes. So you can also resubmit answers for lab 5. Any questions about anything? All right, let me get off the grade book and we'll get started if there's no questions. Oh, there's one other thing I need to mention, and that is you need to get an okay before you begin the infectious disease project. Only about half the students have sent in their infectious disease project. You need to get it okayed by me. And most of the students, I needed to have them narrow down the project. Like somebody said pneumonia, and I told them, no, you can't do it on pneumonia. There's too many organisms that can cause pneumonia. And then the other thing you need to clear it with me is each student has to have a different infectious disease. And so you need to clear it with me to make sure somebody has not taken your infectious disease. Any question about any of that? All right. So let me share my screen here. See if I can find it. Here it is. You should now be able to see my screen. Let's go here. Today is July 26th, and we should be finishing chapter 4 and beginning chapter 5. Let me go ahead and open 4 while we're waiting here. Please note you do have quiz 2 that you need to take by Wednesday. I think it's midnight Wednesday. You can look on the webpage to see when it must be done. Quiz 2 will be the same format. There will be a quiz 2A, which is essay and short answer questions. And then there will be quiz 2B, which is multiple choice. To take the multiple choice quiz, you need to use Respondus Lockdown Browser. Quiz 2 will be covering labs 3 and 4 and chapter 3, as well as the end of chapter 2. Any question about quiz 2 or what we're covering today? All right, let me mention we will have a lab today. And probably what I'll do, I haven't decided, is I'll discuss the unknown project. All right. Any questions about anything? If not, let's begin. Chapter 4. Had we talked about active transport? I think we did. Nobody's responding, so I'm going to assume this is where we are. Where do I have to here? Oh, there it is. So when we're talking about active transport, in all cells, it is the movement of material from areas of low concentration to areas of high concentration, and the cell is pumping those molecules into the cell, and that's costing the cell energy, which it uses from breaking ATP, which then gives the energy needed by the transport protein to move the molecule into the cell. Now, in prokaryotic organisms, we have a special form of active transport, and it's called group translocation. It does require a transport protein to move a molecule from areas of high concentration, from areas of low concentration to an area of high concentration. And then you're moving something into the cell with a active transport protein. So all of that is the same. However, what's different with true group translocation from other forms of active transport is there's a high energy phosphate compound that is used in the process of moving the molecule into the cell. For example, when glucose is transported into the cell using group translocation, PEP, the high energy phosphate short molecule, is used. And then the molecule which is moved into the cell is chemically altered. For example, glucose, which is glucose outside the cell, is moved into the cell with group translocation. and the phosphate molecule from PEP is taken off of PEP and then added to glucose. So you no longer have glucose, it becomes glucose 6-phosphate. It's thought that the reason for group translocation, altering the molecule that it moves into the cell, is if you only use active transport, and let's say you got one glucose molecule outside the cell and you got 10 inside the cell, well, it's very difficult moving the one glucose into the cell where there's already 10. And if you have group translocation and you're moving in glucose into the cell, 5 glucose molecules can move into the cell by active transport, meaning they'll stay as glucose. And then 5 can move into the cell with group translocation, and they'll be converted into glucose 6-phosphate. Now, when we have the last glucose remaining outside the cell, it is much easier moving it into the cell when the gradient is 1 outside and 5 inside than it would be when there's 1 outside and 10 inside. So it's thought that group translocation allows the cells to accumulate substances more easily. Any question about any of that? Remember, group translocation is a special form of active transport that is only seen in prokaryotic cells. All right, any question about any of that? If not, let's move on to the cytoplasm. The cytoplasm is the liquid substance inside the cell membrane. And for prokaryotes, that's the entire inside of the cell. However, for eukaryotes, the cytoplasm does not include the nucleus. So inside the nucleus, it's not called cytoplasm. It's only cytoplasm inside the cell and outside the nucleus of a eukaryotic cell. Of course, prokaryotes don't have a nucleus, so their entire inside is filled with cytoplasm. Cytoplasm is mostly water. It's about 80% water, but it does have dissolved solutes in it, meaning it has dissolved molecules in it. This includes, most of the time, the DNA chromosome, which is dissolved in the cytoplasm. Also, inside the cytoplasm are ribosomes. However, ribosomes are big enough that they do not go into solution. They stay out of solution. And there's a ribosome there. It would be the dots over here in the actual electron microscopic image. Also, the nucleoid region, well, I suppose it depends who you count. The nucleoid region is a region of the cytoplasm where the DNA is, and there's no distinction about where the cytoplasm is and where the nucleoid region is, because it's wherever the DNA is floating, that is the nucleoid region. And we just call it that because where the DNA is stains differently. Another thing that is not in solution inside the cell, although it is, I'm doing the wrong button here. It is in the cytoplasm, meaning the cytoplasm surrounds it, are inclusions. what an inclusion is. Let me see if I mentioned that. What an inclusion is a material the cell is accumulating inside the cell. And whenever the materials become numerous enough, that there's so much of it, that it precipitates out of solution in the cytoplasm, that is when we call it an inclusion. And you can see inclusions in the under the microscope because they're large accumulations of substances the cell has been accumulating. So many molecules are there that they precipitate out of solution.
I had a question. What could be an example of an inclusion?
A cell that metabolizes iron will accumulate iron, meaning it will pump it into the cell. And then there will be so much iron in the cell that it'll start to precipitate out. And then you'll see an inclusion. It's sort of like a lump in the cytoplasm.
Gotcha, thank you.
All right. And it could be many more substances than iron. It's just something the cell is accumulating.
Got it, thank you.
Okay. All right. So I mentioned the nucleoid region is simply where the DNA is. There's no membrane separating the DNA region, the nucleoid, from the cytoplasm. So the DNA can move around in the cell, and the nucleoid region will move as the DNA moves. Because there's no boundary between the nucleoid region and the cytoplasm in prokaryotic cells. I'm not sure why I'm showing you. Oh, ribosomes. I already talked about ribosomes. Ribosomes are where proteins are made in the cell. They have different subunits. We're not going to talk about the subunits like the large subunit and the small subunit. But you do need to know that ribosomes are made up of proteins, ribosomal proteins, and then ribosomal RNA or rRNA. The ribosomes are both protein and ribosomal RNA. You should also know that ribosomes in prokaryotes are smaller than ribosomes in eukaryotes. The ribosomes in prokaryotes are 70S, and the S is just the sedimentation coefficient where the ribosome sediments in an ultracentrifuge gradient. I think we talked about that before. So inclusions, you just need to know that there are reserve deposits within the cytoplasm. There can be different inclusions, and they have different names. We're not going to go over the different names, so you don't need to know those. Just to show you some inclusions, magnetosomes, magnetic inclusions, sulfur granules. This must be the sulfur metabolizing cell. And so it accumulated sulfur in the cell, and the sulfur got so numerous that it started precipitating out of solution. And then metachromatic granules in this cell. Let me blow that up. So there's the cell here in the yellow boundary. And then it has 3 metachromatic granules, which are inclusions. All right, let's talk about endospores. Endospores are a structure that you can see in some bacteria. They're mainly found in 2 families. You don't need to know the families, but Bacillus and Clostridium, and they are survival structures. They are specialized resting cells that can survive many different environments, such as desiccation, heat, chemicals, even radiation. They can survive conditions that the vegetative cell cannot. The vegetative cell is the parent cell that made the endospore. When we have an endospore forming, we call it sporulation or sporogenesis, going through the process of forming the endospore. And then when the environment deteriorates, the parent cell, here we're looking at an endospore inside the parent cell will die, leaving one endospore. So that means one cell will produce one endospore, and then the parent cell dies, and then you're left with one cell. So 1 to 1 is not reproduction. It is a survival strategy. When the endospore returns to the vegetative state, and then the cell becomes metabolically active and can go through reproduction and growth, we call it germination, where the endospore returns to the vegetative state. So the vegetative cell is the normal metabolically active cell that reproduces and grows. The endospore does not reproduce, does not grow. Essentially, it's not metabolically active, but that's not entirely true. It does have slight metabolism, but it's just mostly not metabolically active. And that's why it can survive for years or decades in a harsh environment. When a cell goes through endospore formation, first the DNA replicates itself, and then one copy of the DNA moves to one side of the cell, the other copy moves to the other side of the cell, and then the cell starts dividing. You'll note that when the cell starts dividing, it doesn't happen by normal prokaryotic cell division. What the heck is that called? Sorry, I'm drawing a blank here. Binary fission. It doesn't happen by binary fission where the cell splits down the middle. The endospore will be a smaller portion of the cell. And then the cell will divide into two. The small cell will go on to form the endospore. The large cell is the remains of the parent cell. You'll get two membranes forming around the endospore. And then between the two membranes, we'll get peptidoglycan layer forming between them. And then we'll get a spore coat put on top of the, growing endospore. And then we have our endospore. And the parent cell or the vegetative cell will die, and that will release the endospore. Any question about any of that? There's a little video you can watch on endospore formation. I'm going to skip it now because we're a little behind schedule and it's not that good of a video. All right. Eukaryote cell structures. We have them listed for an animal cell on the right. And then we're seeing an animal cell over here. And then we have a plant cell on the left. And we have the different structures you can find in a plant cell. Realize that both plant and animals are eukaryotes, so they will have a nucleus, as well as membrane-bound organelles, like the mitochondria, right there, or right there. Many of the cell structures are the same in eukaryotes and animal cells. For example, the mitochondria is found in both plant cells and animal cells. You can find flagella in both animal cells and in plant cells, depending on what cells you're looking at. In humans, we only have the sperm cell, which has a flagellum. In plant cells, it can be sperm that has the flagellum, and it can also be a very new plant cell, and it would have a flagella to move around in the environment. And not all plant cells, even when they're just starting, have flagella. All right, there are a few different structures in each, like chloroplasts are a structure only found in plant cells. They're not found in animal cells, although chloroplasts can be found in algae. So that's a protist. But we're not talking about protists, we're only talking about plants and animals. The Golgi apparatus or the Golgi body is found in both plant cells and animal cells. Oh, there it is. The centrosome is a cell structure that is only found in animal cells. And it has two parts. We're not going to go into that. The centrosome is important in forming the mitotic spindle when the cell is going through either mitosis or meiosis in animal cells. And it's curious that the plant cell does not have a centrosome, but it does have mitotic spindles, which help the cell go through either mitosis or meiosis. Any question about any of that? Okay, one other organelle we need to talk about is found in plant cells, but not in animal cells. And that is the central vacuole. It is the largest organelle or membrane-bound organelle that you can find in a plant cell, even larger than the nucleus. The central vacuole is not found in animal cells. However, animal cells have a vacuole, and plant cells have a vacuole or vacuoles, as well as a central vacuole. Any question about any of that? And here we're seeing an animal cell on the right and a plant cell on the left. And this is an actual electron microscopic image of the two cells. All right, this chart shows you some differences between prokaryotic and eukaryotic cells. Let me blow this up, and then we'll go through it. You should realize that prokaryotes are typically smaller than eukaryotic cells, and roughly they're about 10 times smaller than a eukaryotic cells. Now, it depends on what cell you're talking about. So we're talking about the average cells here. Obviously, the chicken egg yolk is the largest cell you've ever seen, and it would be much larger than this, or much larger than 10 times the size of a prokaryotic cell. And you know, I don't know how many times larger the yolk of a chicken egg is to a prokaryotic cell, but you can see the yolk of a chicken egg with your eyes, and you cannot see a prokaryotic cell with your eyes. You would need a microscope or electron microscope to see a prokaryotic cell. There is no nucleus in prokaryotic cells. There is in a eukaryotic cell. There are no membrane-bound organelles in a prokaryotic cell. There are in eukaryotic cells, and there are many in a eukaryotic cell. What they are doing is dividing up the cell into functional units. And for each organelle, we will be doing or performing one thing in that organelle in that cell. Any questions about any of that? Okay. Flagella are seen in both prokaryotic cells and eukaryotic cells. They are both made up of the molecule flagellin. And you don't need to know that the flagella are more complex in the eukaryotic cell. You should recognize the glycocalyx, that some prokaryotic cells have a glycocalyx. And if you can see it under the microscope, light microscope, we call it a capsule. Some eukaryotic cells have a glycocalyx, and we're not going to talk about that. Cell walls are found generally in prokaryotic cells. There are some exceptions. And cell walls are found in plant cells and many protists, but they are not found in animal cells. And if you recall, we include scarlet fever. What the heck is that bacteria? I don't remember what I was going to say, but of course, animal cells do not have the cell wall. Oh, here it is. The cell wall of prokaryotes is chemically complex, much more complex than the cell wall of a plant or other eukaryote. Chemically, the cell wall of eukaryotes is very simple. Like in green plants, all you have is cellulose in the cell wall. And in prokaryotes, you have more than one molecule. The plasma membrane for bacteria is very similar to the plasma membrane for eukaryotes. And I won't have time to talk about that later, but let me mention the reason why it's very similar is that eukaryotes, when they were first evolving, simply used what was available and what worked well. And the bacteria organelle membrane, or the cell membrane, works very well. And so eukaryotes incorporated that membrane into their cells. The cytoskeleton, this is a little bit behind the times. Your textbook author has not gotten it correct, even in the most recent version of the textbook. He was saying that there's no cytoskeleton in the cytoplasm. That's not true. There is a cytoskeleton in bacteria, and we've known about that for the last 15 years. So I'm not sure why your author hasn't updated the textbook on that. The cytoskeleton is found in the backbone of eukaryotic cells, and it's very complex, very pronounced. What else did I want to say about that? Well, the cytoskeleton, it runs through the cell and is responsible for giving the cell its shape. The cell wall typically gives the cell shape, but the cell wall is anchored to the cytoskeleton, and together they make the cell shape. Our ribosomes, we already mentioned, prokaryotic ribosomes are smaller at 70S than eukaryotic ribosomes at 80S. And we'll talk about the smaller ribosomes in eukaryotic cells later in this lesson. The chromosome or DNA also differs between prokaryotes and eukaryotes. In prokaryotes, there's usually one single chromosome, and in bacteria, they lack histone proteins. In prokaryotes, there's usually multiple linear chromosomes, and they are associated with histones. Of course, another difference between the two, prokaryotic cells mostly grow by binary fission or divide by binary fission. Eukaryotic cells grow by mitosis. And then eukaryotic cells can engage in sexual recombination. This involves meiosis. And in prokaryotes, they don't have sexual recombination. The best they have, or the most similar thing they have, is where they can exchange DNA or transfer DNA from one cell to another cell. And usually it's a plasmid that's moving. We'll talk about plasmids a little bit later. Seeing the flagella of different cells, comparing it with the cilia, if there's generally only a few of these structures on the cell, it is flagella. If there's many of these structures on the cell, and these are much shorter structures than the flagellum, then we call them cilia. Both of them are responsible for the cell moving. The flagella allows the cell to swim through a liquid media. The cilia do the same. They all beat in unison. And so on the downstroke, the cell will move that way. All right, both eukaryotes and prokaryotes can move by another means, and that is when they combine with ATP and use up the ATP, we can see the cell moving. Any questions about any of that? So if you take a bacteria and you don't know whether it's swimming or not, you can put it in a semi-solid media, and then the bacteria can swim through the media, potentially going everywhere in the tube. All right, I've already talked about up to cell walls. I'm going to skip that. I already mentioned that the cell membrane is similar in eukaryotes and in bacteria. The main molecule of the cell membrane are phospholipid molecules, but there are other molecules. We're only going to talk about proteins on the cell membrane. The cell membrane allows different things to come into the cell as well as leave the cell. And things can enter or leave the cell by three main transport, what's the word I'm looking for? Transport entities. The first is simple diffusion, where the molecule either moves into the cell or out. From simple diffusion, the molecule moves from areas of high concentration to areas of low concentration. So if the molecule moves into the cell spends no energy getting that molecule to move into the cell. Things can also move into the cell by facilitated diffusion. And as I mentioned, that is the way fructose moves into our gut. And it happens by facilitated diffusion. Once the net fructose outside the cell equals the net fructose inside the cell, there will be no further net change in glucose moving into the cell. You'll note that I didn't word it that there is not any fructose moving into the cell. There's no more fructose moving into the cell when the fructose becomes equal outside the container and the container or solution you have the control in. And then the third way things can move across the cell membrane are by osmosis. Remember that osmosis is the movement of water, a solvent, and then simple diffusion and facilitated diffusion allow the molecule to move either into or out of the cell. All of these ways for simple transport across the membrane happen from areas of high concentration to areas of low concentration, and therefore the cell does not need to spend any energy moving the molecule either into or out of the cell. There is another way that eukaryotes can move material into the cell, and that is by either pinocytosis or exocytosis. Either way is where you're transporting a vesicle either out of the cell or into the cell. And then you can move material across the membrane by vesicular transport, where a vesicle brings material up to the cell membrane, fuses with the cell membrane, and then releases the material outside the cell. And then the reverse can happen where the material is brought into the cell, and then that forms a vesicle when it breaks off of the cell and comes into the cytoplasm. Come on. So for vesicular transport, when you're moving into the cell, it happens by endocytosis. This can happen in two ways. We've already talked about one, pinocytosis, where the membrane starts folding in, where you have the molecules of interest, and that fold in becomes bigger and bigger. And then eventually the membrane reanneals at the other end. And then this is brought into the cell, and the membrane anneals around it so that we bring a vesicle into the cell. And this is called cellular drinking, but the scientific name is pinocytosis. The other way endocytosis can happen is by phagocytosis, where a cell puts out phagosomes. I'll leave that there. And surrounds an object. That's the wrong one. And the phagosome will eventually surround the object and then reanneal to form the cell membrane around the object. And then that brings in the substances into the cell. Let me go ahead and draw that. So here we have our cell. Here we have our substance we want to bring into the cell. Well, the phagosomes will begin growing. Yeah, let me see if I can get the eraser. So the cell membrane is now like this for an animal cell that's putting out phagosomes. And then these keep growing. I'm not going to erase it now, but you're getting the picture. And then these touch and anneal. Anneal is when a membrane comes together to another one, and then they fuse together. Let's make this open over here now. Oops, don't have the eraser anymore. Don't know why I don't. And so this reanneals. And then you can see we have a membrane right here around the substances that we brought into the cell. And then the phagosomes touch each other and then reanneal, forming the cytoplasm outside the substances that we wanted to bring into the cell. And now they're brought into the cell by a vesicle, by a vesicle. Any question about any of that? I'll leave that in case I want to use it in the future. All right, the opposite may happen with exocytosis, where material that we want to bring out of the cell is put inside a vesicle, and we call it a secretory vesicle, because that vesicle will then move up to the cell membrane and fuse with it. So let's go ahead and show that. I thought I had that open. I guess I closed it. So here we have the cell, and here we have a vesicle containing something that we want to take outside the cell. I'm trying to make dots, but they're not quite big enough to see. We'll just do these. No, not quite good enough. This moves up to the cell membrane, and then this membrane fuses with that membrane. Try and draw that here. And then this opens up. We'll go ahead and go like this. And then these contents that were once inside the vesicle are now outside the cell, and they can be released, which actually they are released. Any question about any of that? All right, we already talked about cytoplasm, the aqueous substance inside the cell. The cytosol is part of the cytoplasm, but cytosol refers to the fluid portion of the cytoplasm. In eukaryotes, you can see cytoplasmic streaming, which is not seen in prokaryotes. And that just moves substances around in the, well, inside the cell of a eukaryotic cell. So let's watch a little bit of cytoplasmic streaming. There you go. You see organelles moving around in the cell. That's from cytoplasmic streaming. Any question about any of that? And that is actually real-time photography, meaning it's not sped up. All right. As I mentioned, the cytoskeleton is a framework of proteins that makes the backbone or inside structure inside the cell. And it does give the cell its shape, at least for eukaryotes. There are 3 types of cytoskeleton. I don't know if I ask you any questions about it. Probably not. There's microfilaments, intermediate filaments, which I don't think I have shown here, and then microtubules. You can actually see where the cell is by just putting a membrane around the greatest extent of the cytoskeleton here. And it might be the cell is actually rough like that. It might not be a smooth boundary. Here, actually, the cytoskeleton is almost everywhere in the cell. You can see this is the cell here, one cell. And then that blue region is actually the nucleus. Any question about the cytoskeleton? So microtubules are in green. Actin filaments, which you don't need to know the name, are shown in red. As I mentioned, eukaryote ribosomes are larger. They are 80S in size. They are either free, found in the cytoplasm, and they sort of float around in the cytoplasm. They're not in solution. They're too big to dissolve in the liquid of the cytoplasm. Ribosomes can also be attached to the membrane. They're largely attached to the endoplasmic reticulum, and where you find ribosomes, we call it the rough ER, because the ribosomes make the ER look rough. If you have a ER endoplasmic reticulum that does not have ribosomes, we call it smooth ER. You should note that eukaryote ribosomes are 80S. However, they do have 70S ribosomes inside eukaryotic cells, but only inside the chloroplast and the mitochondria. So in eukaryotic cells, we can find 70S ribosomes, but they are only found inside the chloroplast and inside the mitochondria. The 80S ribosomes can be found anywhere in the cytoplasm. Any question about any of that? And we'll talk a little bit more about this later in the lesson. Eukaryotic cells have organelles, and when I mean organelles, I mean membrane-bound structures. They make compartments in the cell, allowing the different compartments to have different functions. Generally speaking, each organelle is very good at performing at least one function. So each different membrane-bound organelle performs at least one function. Any question about any of that? All right. Now, if something doesn't have a membrane-bound structure around the structure, then we call it a cell structure. But if it has a membrane-bound thing around the structure, we then call it a membrane-bound organelle, or I just use the term organelle. Okay, I think your textbook uses the term membrane-bound organelle and non-membrane-bound organelle. And sorry, I don't use that term. I will call it a cell structure. All right, so those are some different cell structures you can find in plant cells and animal cells. Some of them are membrane-bound organelles, like the mitochondria, the Golgi apparatus, the peroxisome, the chloroplast, the vacuole. But others are not. The centrosome is not a membrane-bound organelle. It is a non-membrane-bound organelle, meaning it lacks a membrane. Any question about any of that? So the membrane-bound organelles have a red box around them in this picture. If there's not a red box around them, then it is a cell structure. Like a ribosome is a cell structure. It is not a membrane-bound organelle. All right, here's a few of the organelles we need to talk about. The nucleus is a membrane-bound organelle for containing and protecting the DNA. The endoplasmic reticulum has two types, the smooth ER and the rough ER. The rough ER has ribosomes attached to it. The endoplasmic reticulum's purpose is to make lipids in the smooth ER and proteins in the rough ER, and then send those ingredients to the Golgi apparatus. The Golgi complex modifies and sorts packages of proteins received from the ER and then sends them out to where they belong. So the Golgi apparatus takes it all in and then may modify the protein and then sends it out to where it's supposed to be. A lysosome is a vesicle similar to a vesicle or vacuole, and it's for storing digestive enzymes. So if the cell needs to digest something, it'll usually digest it with a lysosome. A vacuole looks the same as a vesicle, as well as a lysosome. However, the lysosome digests things. A vesicle or a vacuole may not digest things. They usually do not digest things. If the vesicle is moving, we call it a vesicle, or you can call it a transport vesicle. If, on the other hand, the vesicle stops moving and then just stores the material inside the vesicle in that one location of the cell, this one we call it a vacuole. So initially, most, if not all, vacuoles were a moving vesicle. And then once they stop moving, that's when we call it a vacuole. Mitochondrion is a vesicle we'll talk more about later, but it is the site of ATP synthesis for eukaryotic cells. And in the mitochondria is where we perform cellular respiration, mainly respiration, and that can be either aerobic or anaerobic, as well as oh, I'm trying to blank here. I'm even lost what I was trying to think about now, so I'm just going to move on. Hopefully it'll come back to me and I'll discuss it. The point is, a vesicle can turn into a vacuole. If it is moving in the cell, it's transporting material, and we call it a vesicle. If it is not moving in the cell, it's then storing something in that location in the cell, and that we call a vacuole. As I mentioned, in plants, you can have vacuoles, but they also have a central vacuole, and the central vacuole is much, much bigger than a vacuole. Oh, I know what I was talking about. I was going to talk about the mitochondria generate most of the ATP in a eukaryotic cell. However, in prokaryotes, there is no mitochondria. And so ATP synthesis happens at the cell membrane. We'll talk more about that in a future lesson. Chloroplasts are only found in plant cells. It's the site of photosynthesis. And chloroplasts are only found in some cells of a green plant. What cell of a green plant would you not find chloroplasts in? Come on, somebody's got to know. I'll bet you guys all know. Think about it, and then we'll come back to that. What cells in a green plant do not have chloroplast? Perioxysome looks like a vesicle. We're not going to talk much about it. The centrosome I've already talked about. Let's move on to the Golgi complex. It modifies, sorts, and packages proteins. So all proteins made by the endoplasmic reticulum, if they need to be modified, they'll be sent to the Golgi complex in a vesicle. So the vesicle will move them to the Golgi complex. And the vesicle will actually fuse with the membrane of the Golgi complex. Let me blow this up. So right here, That is a vesicle which is fused through the Golgi complex. The Golgi complex sort of looks like a stack of pancakes, one on top of the other. And then these right here, which we'll talk about what they are, you can call them blobs of butter on top of the pancakes. The vesicles from the endoplasmic reticulum move to the Golgi complex and fuse with it. And they fuse with the Golgi complex on this side or this side. And then the proteins are modified inside the Golgi complex. They can be chemically modified. They may just have something added to it, like a phosphate. I'm trying to think what else you can add to it. And I've drawn a blank, but maybe a sulfate group. And then the proteins are sorted after they're modified. And then they're brought to this side of the Golgi complex, where the proteins will then leave the Golgi complex in a vesicle. And then the vesicle will go to wherever the proteins are needed by the cell. So if this is taking proteins to the nuclear membrane, this vesicle will move to the nuclear membrane and then fuse with it. And then that will bring the proteins to the nucleus. Any question about any of that? Oh, let me state that the vesicle doesn't have to stay within the cell. It can fuse to the cell membrane and then dump its contents out of the cell. And the best way to think about that is a teenager with raging hormones. What's happening is their proteins are being made in their cells, and then they're modified, usually in the endoplasmic reticulum, and then they're brought to the Golgi complex to modify, and then they're leave the Golgi complex in a vesicle, and then those vesicles will fuse to the cell membrane, dumping the contents of the hormones into the blood. Any question about any of that? So there's an endomembrane system in eukaryotic cells. Let me go through it here. Essentially, proteins, if they need to be modified after they're made, they'll be made in the rough ER, because it's the one that has ribosomes attached to it. Those proteins will then be put into a vesicle. The vesicle will then move to the Golgi complex. The vesicle will fuse to the Golgi complex, dumping its content inside the Golgi complex. The proteins can then be modified. They will then be packaged and sorted. And then they'll leave the Golgi complex at the other side as a vesicle. And then the vesicle will move to where the proteins are needed. Any question about any of that? We already talked about vacuoles. Let's talk about the mitochondria and the chloroplasts. The mitochondria is the site of cellular respiration, where most ATP is made in a eukaryotic cell. You should note that the mitochondria has two membranes, an outer membrane and an inner membrane, which is highly folded in on itself to make the surface area of the inner membrane greater. And this is an actual electron microscopic image of a mitochondria. Inside, well, we should mention that the mitochondria and the chloroplast have two membranes, the inner and the outer. Inside the mitochondria and the chloroplast, you can also find ribosomes. And these ribosomes are smaller. They're 70S ribosomes in size. And then another thing you can find inside the mitochondria and the chloroplast is you can find a DNA molecule. Any question about any of that? The same thing with the chloroplast. I don't think we'd use the term thylakoids. I won't ask you about it. If I do mistakenly ask you about it, you can ask for that question to be extra credit. All right. So the eukaryotic cells have membranes around mitochondria and chloroplasts. How did the eukaryotic cell get their mitochondria and their chloroplasts? And that's a rhetorical question. I don't expect an answer. This is what the endosymbiotic theory is trying to address. A long time ago, all we had was bacteria, which will make it a rod here, and a proto-eukaryotic cell, which was much larger than the bacteria. We're calling it proto-eukaryotic cell because it's not a true eukaryote, at least not yet. So proto, oops, I don't have the P in there. And that is, of course, a bacteria, specifically a prokaryote, but we're going to use the term bacteria. Well, a long time ago, the proto-eukaryotic cell, let me see if I can move that. Probably not, because I've already gone away from it. Oh, there, I got it. The no, I don't have it. Shoot. The proto-eukaryote engulf the bacteria. So let's go ahead and, I don't have my arrow here. Go like this. And then for some reason, the proto-eukaryote did not eat the bacteria that it engulfed. So we have the bacteria now inside the cytoplasm of the proto-eukaryote cell. And probably the proto-eukaryote simply kept the bacteria around to eat it at a later time. Any question about anything? All right, so the bacteria was capable of performing aerobic respiration. And I suppose I should say bacteria. It grabbed one glucose molecule from the cytoplasm and then metabolized it. That's not going to be a very good arrow. And we'll try and put glucose here. So the bacteria could make probably about 38 ATP per molecule of glucose. Oops, don't have it spelled right. The proto-eukaryote could only perform fermentation and could only get 2 ATP per molecule of glucose. All right, so the bacteria grabbed the glucose and performed aerobic respiration and then made 38 ATP molecules per molecule of glucose. Now this is what's curious. The bacteria then released some of the ATP into the cytoplasm of the proto-eukaryote. We don't know how many ATP it was, but it was much more the proto-eukaryote said, huh, that's odd. I lost one glucose molecule, but I got a whole lot of ATP molecules. This bacteria that I engulfed is making me ATP, and it's making a lot more ATP than I can. And now the proto-eukaryote said, I'm going to keep this bacteria, feed it glucose, and then it'll make me ATP. And so the two lived in symbiosis. The bacteria got a home inside the proto-eukaryote, and it got glucose that was supplied by the proto-eukaryote. And then the bacteria made ATP for the proto-eukaryote. And the two started living in symbiosis. And now we can actually call this a eukaryote because it has a bacteria in it making it ATP. They have lived in symbiosis for so long now, we do not find the bacteria out on its own any place. The only time we find this bacteria is when we look inside a eukaryote cell. And what this bacteria became is the mitochondria. And now we don't have many eukaryotic cells that do not have a mitochondria. And then there is one family of eukaryotic cells that do not have mitochondria. And what they do is they engulf a bacteria and then live in symbiosis with that bacteria, giving it a home and giving it glucose. And then that bacteria makes ATP for that cell. It's one family of amoeba that does that. You don't need to know that, okay? So we can still see a few, quote, proto-eukaryotes engaging in this symbiosis that we think happened to form the mitochondria. Any questions about any of that? That is the theory of endosymbiosis, how eukaryotic cells got their mitochondria and got their chloroplast. Let's take a look at this and state that in animal cells, they only did this one time, and the bacteria evolved over time, living in symbiosis into the mitochondria. However, in plant cells, this actually happened twice. They first took in an organelle or a bacteria, which became the mitochondria, and then they took in another bacteria, and it became the chloroplast. So in green plant cells, this actually happened twice, if the theory is correct. Let's look at some evidence of this. All right, the first evidence is we do have eukaryotic cells which can engulf another cell and live in symbiosis. And I mentioned the amoeba that is lacking mitochondria lives in symbiosis with a bacteria that it engulfs, and then that bacteria makes ATP for the amoeba, and the amoeba supplies a home for the bacteria and gives the bacteria glucose. This is another example of that, where this cell has engulfed a bacterium and they're living in symbiosis. The protist provides a home for the bacteria and provides the raw elements that the bacteria needs. This is a photosynthetic bacteria, why it's green, and it will make glucose both for itself, and some of it, the glucose, will be released into the cytoplasm of the protist. Okay, so we do see this happening in eukaryotic cells today. Some other evidence is when we look at the growth of the mitochondria, the cells divide by binary fission, which is the way prokaryotic cells divide. But eukaryotes do not divide by binary fission, they divide by mitosis. Some other evidence, the ribosomes in the mitochondria and the chloroplast are 70s in size, and they're 70s in size in prokaryotic cells. In eukaryotes, they are 80s in size. When we look at the first amino acid that is made in protein synthesis, for eukaryotes, the first amino acid is always methionine, but in prokaryotic cells, it's a similar amino acid, but different amino acid, formylmethionine, and it's formylmethionine in the mitochondria and the chloroplast. When we look at the DNA, eukaryotic DNA has histones, proteins on their DNA. The bacteria do not have histone proteins on their DNA. The mitochondria and the chloroplast do not have histone proteins on their DNA. And then I guess another thing is that the chloroplast and the mitochondria have a DNA molecule. They're the only organelles in the eukaryotic cell that have their own DNA molecule. And that DNA molecule makes different proteins for the mitochondria, such as the 70S ribosomes, which in this case is not a protein, that's an rRNA. When we look at the DNA, we have eukaryote. The eukaryotic cells have linear DNA molecules, and there's many of them. There's only one in the mitochondria and it is a circular DNA molecule. And those are the same types of DNA, one usually that's circular. Okay, in prokaryotic cells. We're talking about bacteria mainly. Another thing which is kind of a weak evidence is that if something is brought into the cell from outside the cell, you should see that it'll have a vesicle membrane as well as the membrane, the cell membrane of the cell, meaning something that is brought into a eukaryotic cell should have two membranes. And the mitochondria and the chloroplast do have two membranes. The inner mitochondrial membrane and the outer mitochondrial membrane. And then the strongest piece of evidence, when we sequence the DNA of the mitochondrial DNA molecule or the chloroplast DNA molecule, it's sequenced similar to the bacterial DNA molecule. It does not sequence similar to the eukaryote DNA. So the domain that the mitochondria and chloroplast belong to is domain bacteria. Any questions about any of that? All right, so that is the endosymbiotic theory, a theory to explain how eukaryotes got mitochondria and chloroplasts. Any questions about the endosymbiotic theory? All right, if there's no questions, that's it for chapter, what chapter is this, 3 or 4? Chapter 4, let's begin chapter 5. So chapter 5 is microbial metabolism. This slide is showing you the major goals of this lesson, as well as a rough outline. Know the terms, know what is catabolism, anabolism, activation energy, what is an enzyme, an active site, competitive inhibition, non-competitive inhibition, which is also called allosteric inhibition. What is a redox reaction? What are photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs? Know what is fermentation. And then lastly, we'll talk about both. Well, we won't talk much about cyclic, non-cyclic photosynthesis. We'll briefly mention it. And then we'll talk about cyclic photosynthesis. Any question about any of these terms? So if there's no questions, besides these goals, also be able to understand what feedback inhibition is and understand what aerobic respiration is, its steps, and how it differs from anaerobic respiration. In this lesson, we'll spend most of the time talking about aerobic respiration, but we will talk a little bit about anaerobic respiration and about fermentation. When we're talking about metabolism, you should know that metabolism is all of the chemical reactions occurring within an organism. And because we're talking about microbes, you can say it's all of the chemical reactions occurring within a cell. There are two types of metabolism. There's catabolism, and there's anabolism. Catabolism is where you take a big molecule, like a complex molecule of starch or a complex protein, and then you break it down into smaller molecules, such as their monomers. So catabolism is a decomposition reaction, changing a large molecule into a smaller molecule. Catabolism also releases energy. So it is an energy releasing process. There's more energy in a complex molecule than there is in a simple molecule. So some of that energy is released. Well, actually, all of that energy is released, and some of it may be converted to ATP. Some of it will be converted to heat. Heat is just another form of energy. And whenever you change energy from one form to another, you will always lose some of that energy as heat. So regardless of what's going on, you will always lose some energy as heat. That is a law of thermodynamics. You don't need to know that, but I'm just telling you for your own information. Anabolism is the reverse. It is the biosynthesis reactions where you take smaller molecules, such as molecules of glucose, and you connect them together in an anabolic reaction to make a complex molecule, such as starch. This is an energy-using reaction because starch has much more energy in it than molecules of glucose. So for this reaction to occur, you need to put in energy into the reaction. That energy is usually obtained from breaking down ATP. Once again, whenever you're converting energy from one form, such as glucose, into another form, such as starch, you will lose some of that energy to heat. Any question about any of this? All right, with that stated, let me mention that this slide is a little bit oversimplified, that not all catabolic reactions generate ATP. Most catabolic reactions do not generate ATP. They do release energy, but we cannot grab that energy to make ATP. For example, when we break sucrose down into glucose and fructose, our cells cannot take some of that energy to make ATP. In fact, our cells are very poor at making ATP. Humans only have 2 reactions for making ATP. And we'll talk about both of those reactions in this lesson. It's fermentation and aerobic respiration, which they're not one chemical reaction, but they're separate reactions. They're a process for making ATP. And those are all we can do. Even green plants, which are much better at making ATP than our cells are, most of the catabolic reactions that they perform do not allow them to make ATP. And I don't know how many reactions green plants have, but it's more than 2 to make ATP. But it's many, many less than all the possible catabolic reactions. So most catabolic reactions do not make ATP. However, all anabolic reactions need energy, and they usually get that energy from breaking down ATP. All right, when we're talking about metabolism, a enzyme can speed up a chemical reaction. I think we've already talked about this. We can then hook that reaction from going from A to B to another reaction going from B to C to another reaction going from C to D, and then D to E and E to F. And then we can hook all these together to make a metabolic pathway. Metabolic pathways are a sequence of enzymatically catalyzed chemical reactions in a cell to make the end product from the starting molecule or the starting substrate, which is also called the reactant. Metabolic pathways are determined by enzymes, as the enzyme performs this chemical reaction from A to B. And enzymes are encoded by genes. So metabolic pathways are ultimately dependent on the genes in that cell. Any question about any of that? Now, the last product of a metabolic pathway is called the end product. The starting molecule is called the starting substrate or the starting reactant. All of the intermediate molecules are called intermediate or intermediate products because they can be converted to the next product. Here is a picture showing you all of the metabolism you need to know for microbiology. Easy, right? Only kidding, you don't need to know all those chemical reactions. You do need to know the chemical reactions occur because of the collision theory. The collision theory states that when two atoms, ions, or molecules collide with one another, if they have enough energy, the chemical reaction will occur. The energy that the atoms, ions, or molecules need in order to change from the starting substrate to the products is called the activation energy. The energy needed to disrupt the electronic configurations of the starting substrates and then convert the starting substrate into the product. The reaction rate is the frequency of collisions with enough energy to bring about a reaction. The reaction rate may be increased by increasing the abundance of enzymes, when the reaction will proceed much quicker, or you can increase the temperature or the pressure. If you increase the temperature, the molecules colliding will move about more rapidly, meaning they will gain the energy. That's what temperature is, the movement of molecules. So they gain more energy when you increase the temperature, and then they'll be more likely to collide with enough energy for the chemical reaction to proceed. Pressure happens in a similar way. When you put something under pressure, like when you're cooking food, it tends to put the molecules closer together, and then that makes them more likely to collide, and that can speed up the chemical reaction. Any question about any of that? Here we're looking at a chemical reaction. An enzyme simply reduce the activation energy of a chemical reaction. On the y-axis, we have the energy. On the x-axis, all we have is time. So we have the starting substrate or reactant, and it has a given amount of energy with it. And that is the energy in the chemical bonds of the molecule. You'll note that the reactant has more energy in it than the products. And so this reaction proceeds downhill, and that will give off energy. We'll talk about how much in a little bit. To get the enzyme, the reaction to occur without the enzyme, you need to increase the energy of the reactant so that instead of having this level of energy, it now has this level of energy. And then when two molecules collide, they'll have enough energy to break this molecule apart into the products A and B. And how you can get this to do would be something like you take A and B and you put it under a blowtorch. That will increase the energy in the molecules A and B, which will break the chemical bonds linking A and B together, and then we'll get A and B, the products. That is the activation energy without the enzyme. Now, you can have the same reaction occurring with an enzyme, and you can see that you do need to add a little bit of energy to get this reaction to occur. And that we call the reaction activation energy with the enzyme, from here to here. And that's less than from here to there. And then this reaction can proceed in the presence of an enzyme, and then we'll get our products. One way you could get the reactant to change into the products, assuming that the reactant was sucrose. Actually, that's not the best example. Let's go glucose. And then convert it into CO2 and water. How you can do that without an enzyme is you can take a blowtorch to the glucose and then burn the glucose, give it its activation energy without an enzyme, and that's quite high. I don't know the temperature for burning glucose, but I'll bet it's above 500 degrees Celsius. If you put sugar in the oven, and an oven the hottest it can get's about 500, it'll eventually turn brown, but it doesn't convert into carbon dioxide and water. Okay. So you put it under a blowtorch, and that amount of energy you add is quite high. And then energy is released. That's not shown here, but this is higher than there, so energy is released. And so the energy of a blowtorch is something like 1,000 degrees. And I don't know if that's 1,000 Fahrenheit or 1,000 centigrade, but something like 1,000. To convert glucose into CO2 and water. And yet, in your room, right now, you have glucose being converted into CO2 and water. Can anyone tell me where that is happening in your room right now? Come on, somebody's got to guess. Where?
In our bodies?
Yes. In the cells in your body, we have glucose being converted into CO2 and water all the time. And the reason why we do it is actually to release the energy in glucose and then convert it into the energy in ATP. We'll talk about that later. But the point is, we're burning glucose all the time in the cells of our body. And obviously, the cells in your body are not at 1,000 degrees Fahrenheit, because those cells could not live at that temperature. So the point is, the activation energy with an enzyme is often much, much less than the activation energy without an enzyme. And this is not showing you a true picture, at least for glucose, because you don't need much energy in your cells. You only need your cells to be close to 37 degrees Celsius. And then to convert glucose without enzymes, you'd have to heat the glucose to near 1,000 degrees to get carbon dioxide and water. Any question about any of that? All right, enzymes are biological catalysts. They speed up chemical reactions. They can speed up a chemical reaction 10 to the 8th, 10 to the 10th times. You don't need to know how many different times. You should know, however, that enzymes are encoded by genes. The reason why your cells have enzymes is because your genes code for those enzymes. You should also note that enzymes are specific for changing one specific chemical molecule into one specific product. So enzymes are specific. The specificity is due to the three-dimensional shape of the enzymes. Now that's a little bit oversimplified. If you know work with drugs, we can have a drug which will bind to the enzyme. But for our purposes, it's good enough that one enzyme is specific for binding to one specific molecule. And generally speaking, that is all it binds to. And then it will change that molecule into its products. So the enzyme will not be used up in this reaction. Once the enzyme has made the product, the enzyme will release the product, and then it can bind to the other substrate time and time again. You should know that enzymes form an enzyme substrate complex. And this lowers the activation energy. Let's see, I've got a picture here. Oh, here we go, right here. Ah, that's not a good one. Right there. When the enzyme binds to the substrate, that puts stress on the molecules, the chemical bonds of the substrate. And that's what changes the substrate into the product. Any questions about any of this? All right, I think we'll stop here and I'll continue. Is it at 7 for the lab?
Yes.
Or is it 7? Okay. All right, I'll see you at 7. Let me confirm that.
Thank you.
You're welcome. Yeah, I'll see you at 7 for the lab.